Well, it's great to run into old friends at investor conferences, and I've just done that with my buddy Greg Weaver, CFO at Fibrocell Science, who is here to tell us their story. Good to see you, sir.
Thank you, Robert. Thank you, Ted. The Alliance for Regenerative Medicine, Piper Jaffray, Wainwright, it appears, as well as Maxim. So thank you very much for having us today. And I'm the CFO of Fibrocell Science, based outside of Philadelphia, Pennsylvania, focused on the autologous fibroblast cell as a therapeutic and as a gene delivery vehicle. Forward-looking statements, I will likely make one or two today. So Fibrocell is an interesting business. I've been with the company for about 6 months as a CFO. We're a business that benefits from a couple of really important things. One is a close-knit collaboration with our collaborator and partner, Intrexon Corporation, the leader in synthetic biology. We also have an orphan disease-focused pipeline in preclinical stage, as well as two clinical stage phase 2 trials that are label extension trials on our approved Asfacel T BLA. That's an important cornerstone that enables us to rely on our validated manufacturing site in Pennsylvania to drive follow-on indications, in this case, 2 SCAR indications. All enabled by strong intellectual property and years of experience with the autologous fibroblast cell. We are dedicated and committed to the successful development of therapeutics for rare diseases. Again, we're focused on skin disease and on connective tissue disease. The autologous fibroblast cell, this is what it's all about for us. It's the most common cell in skin and connective tissue. It's producing collagen and matrix proteins and growth factors. It forms the infrastructure of tissue. It lays just below the skin surface. And an important takeaway here is it lends itself to be both inherently therapeutic to treat skin disease, as well as an effective and efficient gene delivery vehicle. So it does both act as a therapeutic, and that's how we're pursuing the SCAR indications, as well as a gene vehicle to talk about regenerative medicine. This is really an important concept for us as we work with Intrexon to treat a rare skin disease by replacing what might be a faulty or a missing gene protein. And we'll get into that a little more here in a minute. Here's how we do it. There's a bit of detail here, but there's the yellow and there's the blue on the right. So on your left, what we have is the Asfacel T, FDA approved through a BLA. Again, initially this was introduced as an aesthetic product. Again, we've shifted the focus to rare and serious skin and connective tissue disease. On the left, the fibroblast cell therapy involves harvesting the patient's own fibroblast cell through a skin biopsy, cleaning it, growing it. Again, these are the patient's own cells. They go through the cultivation and production process in our facility outside of Philadelphia, and the end product is 100% pure fibroblast cell, autologous fibroblast cell, in a 1.2 milliliter vial storage, and ready for clinical administration back into the donor patient. Since the therapy used is person's own cells, it eliminates any risk of cross-infection or rejection. Now, if I introduce on the right side of the cell what we're doing with our partner Intrexon, is the platform provides an excellent opportunity to modify patients' own fibroblasts while they are ex vivo, so that they may, when delivered to the patient clinically, express the protein locally where it's required. And in the case of our autoimmune indications, under the control of an activator ligand. So we're essentially transfecting to genetically modify and force the cells to produce a healthy missing gene, if you will. So we're inserting the missing gene where the protein with the patient's own fibroblast cell. So here's a look at our pipeline. We have the 2 SCAR indications at the top, layered across roughly 2014, 15, and 16. You'll see that these are SBLA programs. Restrictive burn scar and vocal cord scar are currently in phase 2 trials. And we will talk a little bit more about that in a minute. Followed by the green arrows are the 4 preclinical programs that we're working closely with Intrexon on, rare skin disease programs, recessive dystrophic epidermolysis bullosa, or RDEB, morphia profunda and linear sclerodoma, followed by CE, and the most recent addition to our collaboration is the hypermobility tenascin X deficiency. So this is a pipeline that's ambitious, focused on rare and orphan skin disease. So again, briefly, the importance of the BLA of Asfacel T is that it's an enabler cornerstone to allow us to pursue the restrictive burn scarring and vocal cord scarring development, and to rely on our CMC-validated, FDA-approved manufacturing facility in Pennsylvania. This is an unusual and very attractive situation from a risk mitigation standpoint if I'm an investor, is to look at this business not as early stage, but as a company that has a validated manufacturing process, has an approved BLA, leveraging the phase 2 clinical trials, along with the ability to work with the bench strength and the expertise of a partner like Intrexon on the gene modification programs. Let me talk about briefly each indication now. The restrictive burn scar program, this is a condition where a patient has a burn. The skin is essentially, the skin along with any healthy fibroblasts are gone, and it's been replaced by an overgrowth of scar tissue. What we're doing is taking the natural tendencies, remodeling of the collagen with the autologous fibroblast cell delivered to the site of the scar in order to unlock it, if you will, and increase the mobility around the scar tissue at the wrist, the elbow, the shoulder. So we've got a phase 2 trial that is initiated. We've got 4 centers enrolling, 4 more coming online at the IRB stage, 2-to-1 randomization up to 30 patients, 6-month efficacy endpoint, validated measurement scale, but initially the important takeaway here is the range of motion is really what we're looking for. So improving the collagen properties at the site of the scar to improve the mobility around the scar. In the case of vocal cord scar, these 2 phase 2s are running in parallel. Again, the same product, if you will. It's the autologous fibroblast taken from the patient, cleaned, grown, and then re-delivered back, in this case, to the site of the vocal cord scar. The scarring can occur from any number of conditions, overuse, side effect of chemotherapy or radiation, that sort of thing. The autologous fibroblast cells' restorative properties we're going to exploit here, those natural properties, reintroducing them back into the patient's vocal cord membrane. We're going to run the study against saline. Again, there's no standard of care here. And the proposed endpoints at the bottom of the slide here, the absolute change from baseline in the wave grade, and along with 2 secondary endpoints, which are a patient index along with a physician index. Again, this study, again, just came online with the IRB approvals, and we are enrolling patients. Both these trials, we anticipate the enrollment running through Q2, Q3, and results, top-line phase 2 results next year. Let me talk about the Intrexon collaboration. This is a situation where, unlike a typical collaboration, I look at this as much more than that. We're a cohesive team. We're working toward a common goal, and the teamwork is absolutely critical. What Intrexon brings is their industry-leading industrial synthetic biology tools and expertise. Gene libraries, high-throughput screening, ultravector technology, gene transfection and plasmid integration, the Rio switch ligand technology in the case of the autoimmune indications where you want to have a control of the modulation of the expression, and their execution and know-how. That married with the fibroblast technology here at Fibrocell, with our manufacturing and regulatory approved process, again, the common link there, working together to address these orphan indications in skin diseases. So the way the business relationship works is we carve out a field of use, an exclusive field of use from Intrexon. They're paid an upfront license access fee and common stock from Fibrocell, followed by us leveraging their expertise and all these various areas and tools and staffing, and pay them a fee for service, and then some back-end economics through a sales royalty. Here's a snapshot of the size of each market that we're going after here with the gene modification approach on RDEB, tenascin X deficiency, morphia, and CE. RDEB, again, the takeaway here is it's caused by a genetic mutation of the collagen 7 gene. When working correctly, those fibers at the bottom of the slide there in the cartoon, collagen 7, would be connecting the epidermis and the dermal layers of the skin properly. But when you're born with this genetic defect, your body's covered in blisters from childhood and gets worse as you grow older. And it's a heartbreaking disease, and what we're looking to do is treat it by taking the cells again from the patient that can't produce the collagen, introducing a healthy collagen 7 gene from the library. So we modify it and then insert it in and around the area of the wound of each individual blister. It's how it would be administered clinically. So it's essentially each wound is a battlefield, and we'll be working the perimeter with the introduction of the Col7 gene to have the body produce the protein that otherwise wouldn't be there. It's a big deal, and we're going to have proof of concept data around this program later this year. Briefly, we added the tenazin X hypermobility type to the pipeline earlier this year, again working closely with Intrexon. In this case, similar to RDEB, but unlike RDEB, this is the missing tenazin X gene, which results in a hypermobility disorder, which in its worst case can be debilitating and have you wheelchair bound. And what we're doing here is, again, very similar in that we're introducing the gene through the transfection using the autologous fibroblast and then injecting it into the joint where you have a problem. So that's the approach there in preclinical. So that's the stage. And then we also have the RioSwitch technology that is working with, essentially that's a method, again, for putting a specific discrete gene under the control of a ligand. And that's a tool that Intrexon brings to us in order to treat the autoimmune diseases that we have under charter with them, morphia profunda and cutaneous eosinophilia. Essentially, that's a snapshot on the right of a patient with a thick pattern of collagen accumulation overexpressed. And what we'll do is treat the symptoms and treat the underlying disease with our introduction of the control to knock down the inflammation and also the overexpression of the collagen. And as we approach the end of the presentation, I'll mention briefly the UCLA research collaboration we have. Early stage, standalone between us and UCLA. It's been going on for a couple of years, and we're just renewing that for a few more years. There's 2 or 3 different programs underlying that collaboration, but important to note the BMP2 program is essentially where our colleagues at UCLA have transfected a BMP protein gene into a fibroblast cell and have preliminary studies indicate this can be used to promote bone growth and treat things such as bone fusion. And I'll just briefly mention we have a strong balance sheet, roughly 2 years of cash runway. We're using roughly $6 million a quarter. Coverage from a few sell-side analysts, which we support, or which we receive their support and appreciate greatly. And the takeaway on our milestones, the takeaway message here is that we have 2 phase 2 trials in burn scar and vocal cord scar. I'll recap for you here. Those are in phase 2 and anticipate completing enrollment at or around the end of the summer and then moving to reporting out top-line results early next year. And then in the RDEB program, proof of concept animal study completed at or around year-end 2014. We're working very, very hard on that with Intrexon. Some 2015 milestones would include moving those phase 2 trials out of phase 2, and if the results are compelling, into phase 3, along with on RDEB, moving into human trials and moving those other orphan drug indications forward through preclinical and approaching IND. Strong management team, as every other company you see today has, I'm sure, along with a strong balance sheet, which not everyone does have, a market cap of roughly $250 million. I think my takeaway here is that it's been a pleasure speaking with you. For me, I've seen companies that are at or around this market cap size, and in this case, couldn't be more excited about the upside. I'll leave it at that. Thanks for your time. We'll go to a breakout session. Look forward to your questions there. Thank you. Thanks, Dave.